I don't think that our first speaker really needs too much introduction. It is a great privilege to welcome Hans Clevers tonight. He has pioneered and made such tremendous discoveries that reshaped modern biology and science. He is world-renowned for his work on adult stem cells, on development of organoids, and paving the way for these models toward clinical applications. He inspired generations of scientists worldwide, and this group is really growing on a daily basis. So tonight he's going to tell us about the most recent advances in tumor organoid biology, and welcome.
Yeah, thanks very much, Mariana, for a brief but solid introduction. I'm actually MD PhD. A long time ago I saw patients, but it's like 40 years ago. And Navid and Gordana, thanks also for organizing this very timely meeting. I was actually head of Roche R&D till a few weeks ago. I will not talk about Roche programs, but I fully agree with what you just said, Gordana, that actually this field is very rapidly becoming extremely crucial for drug development, particularly because of the stance that FDA has been taken in the past 2 years or so. So what I'll try to do, I have a number of short stories. I give a very brief introduction on the adult stem cell or tissue stem cell based organoid technology that Toshi Sato in my lab started 15 years ago or so. And it was all based on this structure here. You see the inside of the gut. I think I have a laser pointer. No. Yeah. So a mouse has about 1 million of these units. We have about 10 billion. Our small intestine, the colon actually has a flat surface. These are called villi. These are called crypts. There's about 8 crypts or so at the base that flank the basis of a villus, and it's been known for almost a century that there are stem cells somewhere at the base of the crypt that are probably the most active stem cells of the mammalian body because the daughter cells that they produce move up, they divide constantly for about 2 days, they exit the crypt, they differentiate out into multiple different cell types, then they take another 3 days, this is in a mouse, and by day 5 or so they reach the tips of the villi and they die. So this tissue turns over every 5 days in mice, probably more like every week in humans. In essence, you're producing probably about 100 to 200 grams of these cells every day in your small intestine, in your colon, that then demise a short time later. We sort of by chance, when we were still immunologists, discovered that this process is driven by wind. That was probably the first example of wind driving stem cells. At the same time, we found that in cancer, I'll say a little bit about it later on, that actually the same pathway is mutated, activated by mutations. So, and then combining these two observations, Nick Barker found a marker for the stem cells, a wind target gene called LGR5, later on found to be the receptor for a wind amplifying molecule that I'll talk about. And then we discovered that these stem cells don't really fit any textbook description, because people at that time were looking for stem cells as DNA label retaining cells, meaning that they divide very rarely. It was believed that stem cells that do divide frequently are very dangerous because they can undergo mutations, become a cancer, or deleterious mutations would lead to the loss of the stem cell. So these stem cells actually, first of all, are not rare, they're very abundant, and they divide in the mouse every 23 hours. So they go through 1,000 consecutive cell divisions in the lifetime of a mouse, and for us that would be something like 20,000 consecutive cell divisions. The logistics is enormous to get this done, but apparently these cells manage, because colon cancer is common, but it doesn't hit everybody all the time. We also found that these cells don't divide asymmetrically, and there's many other things that didn't really fit the bill that we knew from the bone marrow stem cells. What I'll talk about is using organoids. So we actually managed to grow these cells. Toshi Sato did this. You get small intestinal organoids out. They're driven by these script-based columnar cells, and we now know there's about 12 or so different cell types. There are essentially 6 cell types that make different hormones, including the L cells that make GLP-1, which is basically now exploited to develop these weight-reducing drugs like Mounjaro and Ozempic. So 6 of these in the small intestine. Actually, the stomach has some other ones as well. Pannate cells at the base of the crypt, they are sort of immune cells. They have enzymes here, molecules that kill bacteria. Goblet cells make mucus also to defend the inner lining of the small intestine. Tuft cells, again, a cell that's involved in immunity. It produces leukotrienes, for instance, and can drive out worms out of your intestinal tract. Enterocytes are the only cell type that take up nutrients, which is the main role of the intestinal tract. Best 4 cells I'll talk about, only present in humans, mice don't have them, and then M cells also again involved in immune interaction. So almost all of these cells control metabolism at satiety, but all the other ones protect the gut, and only the enterocytes, the most abundant cell type, actually perform the real gut function. Now, this is a human small intestinal organoid grown by Gu Ge and Lin Lin, 2 Chinese postdocs now leading their labs both in China. For some reason, CRISPR works very well in organoids, so in this case they knocked in a green fluorescent marker in mucin-2 gene, marking goblet cells. You see there are many goblet cells in these organoids. Defensin A5, pannate cells, you see them here in dark, so sitting at the base of what is the equivalent of a crypt, so the stem cells would be in between. Chromogranin A is a common marker for all enteroendocrine cells, these hormone-secreting cells, of which there are 6 types in the small intestine. You can see there's quite a few of these enteroendocrine cells, and actually by single-cell sequencing we can see all of the different subtypes presented in these organoids. And again, you can grow them from a single cell, so it's really a multipotent stem cell that on its own can make a linear version of the gut. When you line these organoids up, they fuse and they make tubes. So first little story is the best 4 cell. With single-cell sequencing, a flurry of papers came out that identified the cell type that histologically had escaped observation by pathologists and histologists. You can see the papers here. They are pretty rare, probably 1% or so of the intestinal epithelial cells. They often sit next to a goblet cell that was reported, but people hadn't really been able to study these cells functionally because there's no cell lines, and rodents don't have these cells. They only occur in, as far as we know, in primates. Turns out that, so here you see this, by the way, sorry, here you see a human single cell plot, and that's where the best 4 cells sit. Mice don't have these cells. And they're defined by a number of molecules. You see them here, a lot of channels and transporters and adrenaline receptors here. And when we look at, you see a whole list of markers. Typically they express genes like CFTR, the cystic fibrosis transporter. This is a chloride transporter that allows water to equilibrate across epithelia. If you don't have the gene, you have cystic fibrosis. Your mucus layer gets very sticky in many organs, lungs, but also pancreas and liver and the gut. But there's other molecules. Guki2C I'll talk about is a similar channel as CFTR. Best4 is something very similar. And it looks like when you look at the gene expression pattern, these cells really are there to control electrolyte balance, so sodium, potassium, proton, pH regulation, chloride, and also with that control water inside the cell and on the apical side of the cell. Also, I point out there's a few transcription factors that appear to be fairly unique to best 4 cells. And they're between small intestine and colon, they're almost identical. In the colon, CFTR, so the cystic fibrosis gene, is not expressed. Otherwise, they're very similar. Now, it turns out we have them in organoids grown under standard conditions. Daizong Wang has done these studies, Chinese postdocs in the lab. Here he has knocked in in CA7 in one of these marker genes for BEST4 cells, TD tomato, and he stains for BEST4 itself. And as you can see, this is the reporter, so you can see this in viable organoid cells, so that nicely stains red, and when you then co-stain with an antibody, you see that BEST4 and CA7, marked by tomato, mark the very same cells. So this reporter works very well, and again you see it's under standard conditions you get 1.5% of the cells in the organoids, something somewhat similar to what you see in vivo, have these cells. So we now have live BEST4 cells. By single cell sequencing, they look identical to what you get from an adult gut. This is, I should stress, somewhat different from iPS-based organoids that tend to often represent late fetal stages. In these types of organoids, because you start from a fully specified stem cell, actually you typically get the mature adult stages when you take the growth factors out, because when the growth factors are in, they tend to be a little bit younger and often they're still sort of proliferative. Dijsselbloem asks, how can we actually manipulate the numbers of these organoids? We've learned over the years that by changing growth factor conditions, we can bias the production of cells very strongly, sometimes to like 100%. Here he's been adding a number of immune regulatory factors, interleukins, chemokines, and it turns out that interferon gamma, and particularly, as some others do a little bit, but gives a very strong induction of the CA7 marker, so of the number of BEST4 cells. So it goes from 1% to almost 10%. You see here the markers that he used. And then when you visualize this, so this is an organoid grown under normal conditions. You can see these slender BEST4 cells. They have the same shape as they have in the gut. In green, you see goblet cells. So these are the cells that make mucus in the gut. And then we add interferon gamma, and you see a massive increase in these BEST4 cells. This also happens, meanwhile, we know, in vivo. So they are involved in electrolyte balancing, water balancing in the gut, and they are really responsive to interferon gamma. And interferon gamma is particularly produced in the gut upon pathogenic bacterial infections. He then realized, Dijsselbloem, that we can also create organoids that don't have BEST4 cells, and simply you basically remove SPI-B. SPI-B is a transcription factor. The next story also has it. It's a transcription factor very well known to immunologists because it's essential for creating B cells, macrophages, and dendritic cells. So it's a little bit unusual to see this in an endodermal derived tissue because it's really an hematopoietic transcription factor. It also says something about the fact that all these endodermal cells still perform an immune function. Anyway, you knock out, there's 3 of these transcription factors that he found. If you knock out SPI-B, all of the BEST4 cells are gone. Has4 or MEIS1, you knock them out, you see no effect. So apparently SPI-B is key for setting up this lineage. The other ones are redundant or have no role. You can see basically there is not a single BEST4 cell in these organoids. So then combining these tools, he then asked, well, he had a hypothesis. So could it be that these BEST4 cells essentially are our defense against pathogenic bacteria by allowing the gut to produce massive watery diarrhea? CFTR is known to transport chloride, but also known to allow the transport of a lot of water outside from inside the cell to outside the cell. BEST4 does something very similar, so that's the marker of this particular cell lineage. GUCI2C, already mentioned, is a receptor for GUCAs, small proteins. When it gets triggered, it produces cyclic GMP, and both cyclic GMP and cyclic AMP will open these channels. And for instance, cystic fibrosis, what happens if you open that channel, you get massive export of chloride ions and water. There's an assay actually for cystic fibrosis that we set up in Utrecht a number of years ago. Basically, we make organoids that look like this. We raise cyclic AMP in these organoids by adding cholera toxin, a toxin that's known to raise cyclic AMP. The channel opens because it's under control of cyclic AMP, and the organoid swells. And this is now used in a number of European countries as a personalized test to know if Orkambi or one of the Vertex drugs that have been developed will or will not work in an individual patient with this disease with a rare mutation. That patient will that mutation has not been in trials, but it turns out that this assay for CFTR function is black and white. If the organoid test is positive, the patient responds to the drug. It's probably the first personalized medicine-based approach for organoids. So Daizong asked, is this actually what these best 4 cells do? We should probably also say that all of these markers are expressed in mice, but in mice they're just on the enterocytes. So this 90% of all the cells are enterocytes. They all have these transporters, and apparently in mice you control electrolytes and water through the enterocytes. Humans, the enterocytes do not express these genes, but this is a specialized enterocyte. It's a derivative of the lineage, and apparently we, for some reason, concentrated this entire function in a rare cell type where we can increase the numbers by gamma interferon when there is a bacterial infection. Now, and then the test, this is not the best movie, I should say, but essentially what you see here, every second is an hour. So these organoids, when you open these channels, they swell very rapidly. So this, we don't add cholera toxin here, so that would actually do this for cholera. But this is a peptide that comes from E. coli, a pathogenic E. coli, that made this heat-stable enterotoxin. And as you can see, when we add this peptide, we activate CookA2C. This is this receptor I pointed out. Cyclic GMP is produced, the channels open, and the cells produce a lot of water that enters the lumen of these organoids, which is equivalent to the lumen of the gut. So from this, and then the spike B knockout, that has all the other cell types in normal numbers, but doesn't have best 4 cells, it doesn't swell. So the swelling, the function of all of these transporters in the human gut is strictly limited to best 4 cells, and so we postulate now that watery diarrhea, which is a defense mechanism against pathogenic bacteria by just flushing them out of your intestinal tract, that actually the best 4 cells have been specialized to do exactly this. They probably do other things, and we're still trying to study these cell types. So this is, I guess, an example where rodents would have never told us anything because rodents don't have these cells. Another example, M cells. So again, this is the gut. Villus, these would be crypts, so stem cells would be somewhere here. Paneth cells are here. If a daughter cell doesn't move up a crypt, but it moves over these sparse patches, so these are like lymph node-like structures in the gut, the enterocytes now specialize to become so-called M cells. And it's known in mice, so mice have these cells, that this is controlled by a ranked ligand that's secreted by these immune cells, picked up by the differentiating epithelial cell here, that then by the time it reaches the top of this dome is a full-blown M cell. And M stands for microfold, so rather than brush borders, this is not a good image, rather than brush borders they have a few microfolds, and they're very different in the end by EM from enterocytes. The textbook function of these cells is they take up particulate parts of luminal contents, stuff that's in your gut, they transport it by transcytosis, hand it over to dendritic cells, and this then helps control immune responses in the gut, and probably very important for developing tolerance, but also for developing immune responses to pathogenic bacteria. So they're not an immune cell in their own right, but they take up antigenic potential antigens, transport them into these. There's a lot of literature in immunology where dendritic cells are believed to stick out fingers between these M cells. We have never seen that, or elsewhere in the intestinal tract. So we believe that M cells really are specialized to bring antigens in rather than that the dendritic cells reach out. This is an anticipation, but they are quite unusual. So you can see that the enterocytes have these well-developed brush borders. M cells don't have that. Inside M cells, you typically see immune cells, dendritic cells, B cells, T cells, 3, 4, 5 cells occasionally. We don't know how they get in. Probably this is an opening here, probably from the bottom, because they come from below the epithelium. We don't know what they do there, so we're trying to hope actually to set up a system that we can study this in organoids. This is again Daizong. He again played his trick of trying a lot of soluble factors out, and he finds out that ranked ligand in humans induces organoids a little bit, 2% here. If he then adds GM-CSF or CSF2, you can see we go up to about 10% of mature M cells. And the sole marker in the literature for M cells currently is a transmembrane protein called GP2. Now, with this system in hand, and also with a lot of the published single-cell sequencing databases, data sets from healthy and IBD guts, for instance, he could put together a differentiation trajectory, so that the stem cells would be somewhere here. The enterocyte precursors that happen to travel over a porous patch will become early M cells. Actually, it's the same transcription factors again, SPI-B. SPI-B, this immune transcription factor, so best 4 cells have it. M cells also have it. We actually found this a number of years ago. And if you knock it out, you make no M cells in organoids. By EM, you can nicely see enterocytes with their brush border and M cells in 3D organoids. In 2D organoids, we also do a lot of 2D cultures nowadays. Staining for GP2, so the fully mature marker here, you can see individual M cells in between these much more normal enterocytes. So now you look on top of a layer of what would be over a porous patch. And the trajectory is quite simple now. So cells start by expressing SPB. They then express a surface marker, ICAM2, that allows us to sort out the immature M cells. Actually, many more of these. They actually look like they're functionally already mature. They lack a few of the markers, including GP2. And then finally, they're GP2 positive, and that's when they now will allow immune cells to sit in this cavity inside the cell. So now we have organoids with live M cells, and M cell field has been very complex because mice have a few thousand of these cells. Human cell, there's no cell lines, there's nothing that you could actually study them in human, but now we have live human M cells in the lab. Here we noted, after we looked extensive at mRNA-seq data, that they express constitutively very high levels of class 2 MHC. Now, if you're an immunologist, you know that class 2 MHC is typically expressed by antigen-presenting cells, so macrophages, monocytes, and B cells. Very important for the uptake and presentation of exogenous antigens to T cells, to the immune system. Turns out that, so if we do on these organoids, when we induce M cells, we stain on the x-axis for GP2, so there's the 2% of mature M cells. But if we stain for class 2, there's many also immature. So these are immature M cells and these are mature M cells. Without the GP2, so these are antibody controls. Normal intestinal organoids that have no M cells have no class 2 positive cells. So very important. So the normal epithelium doesn't do this unless we give high-dose interferon, and now all of a sudden we induce a lot of interferon in enterocytes. And this is actually what many people have been studying in the context of celiac disease. This is the gluten insensitivity. I'll get back to that. So how come that M cells constitutively express class 2 and enterocytes need interferon gamma for that? It turns out that they also express a transcription factor that was very famous when I was still an immunologist. This is a gene cloned from Behr lymphocyte syndrome patients. So these are patients that cannot express class 2, cannot present antigen, severe immunodeficiency, because they lack this particular transcription factor, C2TA. Now, if we knock out, so they express C2TA, if we knock out C2TA in these organoids, we lose all class 2 expression. So they again behave exactly like antigen-presenting cells. They express class 2 constitutively under the control of the antigen-presenting cell transcription factor. When we then look carefully at their unique marker genes, there are many genes here that are expressed by dendritic cells. Now, dendritic cells are probably the most professional antigen-presenting cells of the mammalian immune system. Of course, the immune system is hematopoietic, is mesodermal, and we are talking here at endodermal epithelial cells. We're not talking at the offspring, not looking at the offspring of M cells, of bone marrow stem cells. We then wanted to test if these cells actually are real professional antigen-preventing cells, because it looked from the gene expression profile, they have class 2, they have invariant chain for the immunologist, they have a lot of the the CD80, CD86, etc., etc. So can they actually take up antigen and present it rather than transport it into these porous patches to the real dendritic cells? So the only really well worked out antigen system in the gut is for gluten, is celiac disease. particularly this peptide here. So this is the native sequence as it occurs in gluten protein. Gluten proteins, as you probably all know, are proteins in wheat and rye and barley. There are multiple peptides that will lead to celiac disease. So this is a very serious intestinal disease. It's not just your gluten intolerance in a restaurant. It's you have no villi, you're very sick, you lose weight as a kid, etc., etc. It has been found quite some time ago that this is not the antigenic peptide. The cells that take up the gluten protein and process it actually express transglutaminase, and they turn every Q glutamine into a glutamic acid. And this is the antigenic peptide that's presented. Now, who expresses this enzyme in the gut? It turns out that the M cell has massive amounts of transglutaminase too, and the enterocytes, where all the mouse models have concentrated on, essentially don't express this enzyme. So they cannot take up gluten. Of course, also they're not built to take up antigens, whereas the M cells are. So then finally, could we actually show that when we give peptide or when we give whole gluten protein, this slide gives the peptide, but you can do the same thing with whole gluten proteins. When we make it soluble, you have to do some proteolytic processing for that. We don't give transglutaminase. That's actually in the cocktail that you can buy to study celiac disease. So we have M cells in our 96 well plates. We have reporter T cells, or we have an antibody that can see class 2 MHC with the peptide. And then we can measure either binding of that antibody, or we can measure T cell activity. This is binding of the antibody. So here you look at M cells. These are the M cells that actually have been given the peptides or have been given the proteins. GP2 is the apical marker, so this is where the antigen comes in, and this is the staining this is with an antibody that sees the risk MHC class 2 allele DK2.5, so this is the allele that's associated with celiac disease that can present this particular peptide I just showed, and the antibody that we use here recognizes this as if it's a T cell receptor, so it sees the peptide in the context of this class 2 molecule, and it nicely stains here. There's a lot of controls, of course, involved that I don't give here. And then finally, you can also read this out with a T cell that actually has a T cell receptor to see this class 2 MHC with this peptide. And as you can see here, this also nicely works. This is a Jurkat cell line with a particular T cell receptor that I don't think is named in this slide. Anyway, so from this we conclude that M cells, human M cells, act like professional antigen-presenting cells. Probably the first example outside the hemobiotic system that does this, and it's probably going to be very relevant for celiac disease. And again, this is so people who study celiac disease in animals studies mice. But in mice, M cells don't do this. They don't have class 2, they don't present antigens. So this again would be a reason to not only work with mice, but also work with humans. This paper is going to be coming out soon. Briefly, some older remarks about growing organoids from cancers. I guess so iPS-based organoids that we hear a lot about are really good in building tissues where there are no residual stem cells, like brain or retina or heart muscle or podocytes in kidneys that have no self-renewing function. But this particular technology is really good in growing cancer, so you can grow healthy and diseased cancers, you can sequence them, you can make biobanks, and then also perform drug screens or other experiments directly on cancers that they actually have derived in a personalized fashion. Now, we've made many of these biobanks in Utrecht. This is what they look like, where we have matched wild type and tumor tissue growing as organoids side by side. So the only difference here would be that you have the same genetic background, but then this one would have all the oncogenic mutations. So a number of unexpected observations with these organoids, two essentially. One is that these grow slower than these. So you would think that cancers are really good in growing, they're really not good in growing. I'll show that in the next few slides. And if you have a mixed sample, if you get a tumor sample from the clinic and there's some wild type cells in there, they will overgrow your tumor. So in 2-3 passages, and I'm sure there are multiple papers published where people were thinking they were studying cancer cells, but actually were studying wild type cells, and we've been fooled by that as well. That's one observation. The other is that actually when you now do drug testing side by side, typically these cells tend to be more sensitive than these cells to the typical approved cancer drug, explaining why there are so many side effects. So cancer drugs are really not cancer specific. They've never been developed in a context where there was also wild type tissue available. People claim normal cell lines. They don't exist. Normal cell lines, when you transplant them, they make cancers. They make tumors. So in these tissue, when you transplant these organoids, you actually make normal tissue. This has been done by multiple labs now. So tumors are bad growers, and cancer drugs basically kill normal cells at least as good as they kill cancer cells. And this shows a little bit why they why tumors are poor growers. What you see here is a normal organoid. This is an H2B, GFP, I guess, cell line. So you see the nuclei, and you can nicely see cell divisions. There's going to be one here. And every cell division results in 2 healthy daughter cells. And we typically see this in all normal tissues. If under optimal growth factor conditions, every cell division gives 2 daughter cells. And you have to realize this is not the normal tissue in its normal state. This is the normal tissue in a regenerative state. So we basically maximize the proliferative capacity in this tissue. When we take the growth factors out, they stop growing. If you now look in what happens in a cancer organoid, so this turns out to be a single cell with 2 nuclei. When it goes into a mitosis, you can see where here it happens. It probably has something like 100 chromosomes, far too many. Tries to segregate them into 2 nuclei, doesn't really work very well. There's another attempt here. 3 nuclei, doesn't work very well. There's another attempt here. Essentially the 2 daughter cells die because the chromosomes are segregated in the wrong way. And this is typically what we see in virtually all solid cancer-derived organoids, is actually that they're like, the lab calls them zombies, so they cannot sprint. They basically struggle on, but and pieces fall off, but they just never stop. The wild type organoids can grow very, very fast, but the moment you take the growth factors out, they stop and they differentiate out and they make normal tissue. So we think what happens with many cancer drugs is you kill a lot of the normal cells, you kill a lot of the cancer cells, but the normal cells that are still there come back much faster than the cancer cells. And that's the reason that there is some specificity of these drugs for cancer. Yeah, and then one quick application. So this was one of the early colorectal cancer biobanks we made, and there was one case here that didn't have any mutation that we could see in the EGF receptor KRAS pathway. So now side by side we could take that line and then take a derivative of that line where we introduced a point mutation in the KRAS. So there's only a single base difference between this one and this one. Proof of concept, we added 2 inhibitors here that should kill a KRAS wild type organoid. You can see it nicely kills it, and as already published multiple times, that KRAS mutant should actually block the effect of these drugs. And you can nicely see, actually they go into cell cycle arrest, but they don't die, and then when we take the drug away, it happily grows on. And this is exactly what you would see in patients. So this is one use of normal organoids. You can start from normal organoids and give them 5 oncogenic mutations, turn them into a full-blown cancer. You can also revert, you can take a mutation out with CRISPR and see how that affects a drug response, for instance. But this is, here I should declare that I have some personal involvement. This is a collaboration between my lab in Utrecht, a company called Meres that's developing this antibody now, lab of my former postdoc Edu Batje in Barcelona, and another small company in Utrecht. And what this is actually, I think, the first example where a drug was developed entirely on organoids. And what we did side by side, so and remember, so one, it's a bispecific antibody. One arm is EGF receptor binding. The other arm is LGR5 binding, our stem cell factor. We made a library of about 500 bispecific antibodies, where one arm was a stem cell marker, LGR5, LGR4, RNA43, ZNR3, there's many, and the other arm was an EGF receptor family member. This library of about 500 was screened on, where is it? I hate these slides. A small panel of 3 specifically selected cancer organoids, colon cancer organoids with defined mutations. also the wild type counterpart from the same patient. So we had 3 pairs of wild type cancer, wild type cancer, wild type cancer, and we screened these 500, and we wanted to find drugs that would, or bispecific antibodies that would leave the wild type tissue intact and would only kill the cancers. So I don't think this screen has ever been done before. And that actually in the first screen we got a few hits. We then extended the organoids to a much larger biobank, more capturing the diversity of this disease, and eventually ended up with a single antibody that seemed to be able to kill in a cancer-specific fashion, and also surprisingly it kills KRAS mutant cells as well, whereas EGF receptor antibodies will not work in KRAS mutant cancers because it's upstream from RAS. And that antibody is now actually, it went from, I know how slow this process is in Roche, this went in 2.5 years from an idea to a phase 1 trial. It is now in phase 3, it does exceedingly well. This is actually in head and neck now, and particularly because it turns out to be extremely safe. It doesn't give the particular side effects of EGF receptor antibodies. So we feel that this is NS1, but this actually makes the argument that screening in wild-type organoids and in cancer organoids probably leads to much safer drugs that are cancer-specific and leave wild-type cells, at least to some extent, leave them untouched. And then finally, as probably many of you know, Matrigel is expensive, and Matrigel is quite different from batch to batch. Matrigel is animal-derived. It's produced by growing a mouse tumor in a mouse abdomen. First of all, it's not animal-free, but then also the product is laminin 111, collagen 4, mostly laminin 111, collagen 4, fibronectin, and hundreds of other proteins, including growth factors and all sorts of other biologically active molecules. Definitely not something that can be developed to be used in the clinic. So to make this clinical grade would be very hard, but also not ideal for industrial use. Also the fact that these 3D cultures, that's what I learned in my stint in Basel, not very attractive if you develop drug. If you want to do a high throughput screen with 1 million compounds and you have these 3D things, also because organoids all have different sizes, it's very noisy if you start measuring. You never know how many cells there are and what context they grow. There's this gel that gets in the way. So we, but also many other labs have been trying to look for alternatives for Matrigel. So why is Matrigel so key? Well, this is an epithelial cell. They are polarized, and they always reside, whatever epithelium it is, ectodermal, mesodermal, endodermal, it always resides on the basement membrane. The basement membrane contains laminins, collagens, etc., and Matrigel is apparently a very good substitute for the basement membrane. I should say that laminin 111 is a fetal laminin. It doesn't occur in our bodies as we sit here. That's more like 511 or so, but it does something similar. And the idea is that actually integrins that sit here will allow an interaction with these extracellular matrix molecules. And the signal that's then generated will first of all allow the cell to polarize, so then it knows that this is the top and this is the bottom. This is where my brush border should be if I'm an enterocyte. This is where the hormones should go if I'm an enteroendocrine cell. But maybe more importantly, this is also a signal that prevents anoikis. If you take an epithelial cell, unlike an immune cell, if you take an epithelial cell out of its contact, it dies in a matter of hours, unless you add Rho kinase inhibitor, which mimics integrin signaling, or unless you have some sort of a basement membrane-like substance around this cell. Integrin signaling, I'm not an expert here, but I try to explain. So integrins, alpha chain, beta chain, is believed, so the green one is the beta chain, is believed that the beta chain is the most important one for outside-in signaling. But the first step is actually an inside-out signal, so something happens inside the cell, some other activation step. This takes the alpha-beta dimer from this inactive band conformation to what's called a primed or an extended conformation. And now it develops an affinity for its ligands. This can be collagen, laminin, fibronectin, but there's also surface molecules for some of these alpha-betas. And then when this open product interacts with collagen, for instance, if it's alpha 1 or alpha 2, a signal is an outside-in signal is generated, and then particularly when clustering occurs, this becomes much stronger, and it's these signaling mediators that set up polarity and that prevent anoikis. And that's presumably why Matrigel works well, because Matrigel can eventually take integrins from here all the way to here. Organoids, so this is gut organoids, but we see this in most. They abundantly express beta 1, so that's a signaling chain, and then they express, we think functionally the most important one is alpha 6, which is a laminin. So alpha 6 beta 1 is one of the laminin receptors. Alpha B beta 1 would be another one. And indeed, when we knock out, when we knock out alpha 6, basically we cannot grow the organoids any longer, but when we give them the Rho kinase inhibitor or something similar, they will grow. So that's how we can grow these mutants, but as soon as we place them in a normal environment, even if they put them in Matrigel, they will die. So it looks like indeed it's the integrins, alpha 6 beta 1 is the key for gut organoids that allow Matrigel to work. And of course, there have been many attempts to use so-called functionalized hydrogels with RGD peptides, RGD mimicking a key part of fibronectin, which is the alpha 5 beta 1 integrin binder. We have tested hundreds of hydrogels, and there's essentially nothing has allowed us to grow organoids long term without much change. We could sometimes keep them alive for 1 or 2 or 3 passages, but not 15 or 20. So we've also had many attempts that didn't work. We then turned to bacteria, and Helicobacter is presumed to express a molecule that binds to integrins and activates them. We tested it, we couldn't get it to work. But then Joost Wijnakker, a PhD student in the lab, turned to Yersinia. So Yersinia is a genus, Yersinia pestis. The plague is one of them. They have the gene that I'll talk about, but they have a frame shift in the gene. So the important part of is encoded, but is not produced. But there's several other enterocolitica and pseudotuberculosis that also have the gene that I'll show you now. So what you see here again is a Paris patch, M cells. M cells, already told you, they have class 2, but for some totally unknown reason, it has been known for a long time, integrins of M cells don't sit on the basal side, but they sit on the apical side. And it's the only epithelial cell that I've seen that have their integrins on the wrong side of the cell. It's unclear why they have this. But this is exploited by Yersinia. So here you see the integrins. This is not Yersinia yet, but Yersinia on its surface, you see here's a bacterium here, on its surface has these molecules, they're called Invasin. It's a single chain molecule where the C-terminal tip can actually bring together an alpha and a beta, activate it in such a way, and it has to cluster, that now the integrins get activated and they essentially transport the bacterium straight through the M cell into the underlying tissue. And this essentially is the key step in infecting mammals or human beings with Yersinia. A lot of work has been done on Invasin, particularly by Ralph Isberg in the 90s. These are actually incredibly beautiful papers in Cell and in Nature on the structure, the function, and the immune system, etc., and then totally forgotten. It turns out, so this is the C-terminus, it's on the outside, so it's a type 2 transmembrane molecule. This particular part, 20K, is the business end of this molecule, so the 20K fragment on its own will bind and activate integrins in Ralph's papers. So Joost had read this, wrote a paper to Ralph. This protein is also extremely stable. You have to realize it sits on the outside of Yersinia, in the gut with all the enzymes that are there and all the bacteria that are there, and it survives in the gut. So indeed it's extremely stable, because Joost asked Ralph, Ralph is sort of more or less retired, he's at Tufts. He still had a tube that was like 25 years old in the freezer, and he sent it up and to Holland. Sorry, a little bit more. So Invasin in the literature from 2 or 3 decades ago shows that it doesn't bind collagen receptors, but it binds all of the alpha betas that you see in this red square here. So importantly for us, alpha 6 is here, alpha 7 and alpha 3 are here, laminin receptors. The fibronectin receptors, alpha 5 is very important, but also alpha V. And then this is alpha-4 surface molecules is more important for immune cells. Anyway, so Invasin can bind and activate multiple integrin complexes. So the question is, can we actually use this in culture? And it turned out to be exceedingly simple. What you see here is Joost has plated out a single stem cell from an organoid line that carries I think it's called the Fuji reporter. So cells in G1 are red, cells in G2M are green, and S-phase cells are yellow. You plate a single epithelial cell out on plastic, coated. So it's a simple coat, an hour PBS with Invasin, you wash it off and you put your cells on. This cell would have died right away if it was plastic or albumin or something like that, or any other coat, but you can see it nicely grows into a clone that comes up. BME can support, BME's Matrigel can support this as well, but Invasin support is much better. When we now let them grow from multiple passages, and we let them polarize, you can see here, if we do this on 2D for Matrigel BME, you get polarization, but on Invasin the polarization is much better, as you can see here. And these are mature enterocytes with a mature brush border stained for villain. And here you see it in 3D, indeed, the whole sheet is fully polarized. And I saw another lab using this now last week, where you now, because of course you don't have the round structure with the buds where the stem cells sit, but in these sheets there are fields where the stem cells sit, and basically the daughter cells grow out from in this sheet. Of course, an epithelium is always a sheet, so it doesn't have to be folded up in 3D and assist. It can take any shape. So the fully polarized, we can actually do 12, 15 passages, they're fine. If you do single cell sequencing on 2D or 3D, you see that here, or Invasin, Matrigel, or primary tissue, you get the exact same signatures for the cell types. This is the same organoid I showed in the beginning. In green, you see goblet cells. So they basically secrete their mucus to the top. These are Pannet cells. They have the nice shape, and what you see very nicely here, these enteroendocrine cells. So some of them actually have receptors that would see something in the gut. They have this bottle-like shape, exactly like they would have in a real gut, and they secrete their hormones into the bloodstream, so to this side. So you can nicely see the bottle-shaped EECs, enteroendocrine cells. And again, BME compared to Invasin, if anything, Invasin is at least as good and generally tends to be better than Matrigel. This is all 2D. Structure here, Ralf Isberg had solved the structure on Invasin, but not a structure in the context of integrins. This is done by Tim Springer, who we have been collaborating with in Boston, and his postdoc Saket Bagde and Jing Li. Of course, there are structures of laminin bind to integrins and fibronectin bind to integrins, and actually it's the exact same amino acids, I don't show this here, that are interrogated by this yellow protein Invasin, but the surface of contact is much higher, so the affinity is much higher than what you would normally see for the extracellular matrix proteins. We published this, but it's now an organoid sheet. Now people say they're no longer organoids. I must say I've always thought that an epithelium is a 2D structure. It's not a 3D structure. And two epithelial cells next to each other don't know whether they're in a sheet or in an organoid, right? It's going to look like this. But to show that actually this works in 3D as well, we collaborated with Paul Kouwer in Nijmegen in Holland. This paper is coming out. So Paul has developed a gel, a hydrogel, polyisocyanate gel. I'm not a chemist. That has the same temperature behavior as Matrigel. So it's liquid at 0 degrees. It's a gel above 20 degrees. Also, click chemistry. I have no clue how that works. But you just add protein and it binds, is what we understand. So we basically add our Invasin to that gel. It makes a gel. Typically, when you overload, when you couple too much of the Invasin, you lose a bit of the gel-forming properties, but actually the optimal dose of binding works well for what I'll show you. Also, this gel you can actually take back and forth between gel and liquid multiple times. It's fully transparent, has no color, so it's really, I think, a nice gel to work with. Here we go an organoid this is in Matrigel here, in BME, and this is the same organoid grown in in Invasin in 3D in these PIC Invasin gels. You can see again the polarization is even better. Cells grow very well. We've done multiple passages again. When we do this straight from, it's a bit of a messy experiment, straight from tissue from the mouse where LGR5 has been marked by GFP, by a knock-in, so that you see the stem cells. This is exactly what we would see. This is BME, this is Invasin again, so we can use primary. Meanwhile, we've done many cancers, so primary samples, grow them in 2D or in 3D, and they will grow. And then finally, this is again this reporter with the purple enteroendocrine cells, the green goblet cells, and the orange pennant cells. And again, we get all of the cell types. We've grown these, I think, for 8 passages. Then did single cell sequencing, compared BME to Invasin and to primary tissue, and again, they look identical. So we think that Invasin is probably, we'll make it available for any academic researcher. There's quite a few labs that are working with it already. There's no further claims other than that you don't hand it over to people that we don't know. Companies will have to take a small license. We believe that this might be a way of standardizing. The 2D will help the standardization in the industry, where you can now do 384 well plates, use the robots that people use in industry, et cetera, et cetera. It's probably much easier to standardize and to get better statistics, because you can add exactly 5,000 cells or so per well. And 3D, I think it works probably as well as Matrigel. And with that, I think I named everybody who was involved in the studies, and I hope I left some time for questions. Thank you very much.
Any questions? Well, I can start. So you mentioned first that these best 4+ cells are in the gut, uniquely human, respond to interferon gamma. And I'm just wondering what you know about them in Crohn's disease, in inflammatory bowel syndrome disease, in any kind of autoimmune conditions that might affect the biology of cells.
We've looked at that, so we can of course probe data sets that are online, but also we've done quite a bit of histology. What you see generally when there's more inflammation, you see a lot more of these cells. But histology, we can't really do any experiments anymore, but it looks like indeed, whenever there's gamma interferon produced, the numbers go up, and in inflammatory situations you'll see that, yeah.
And do these M cells that are, what I understood, antigen-presenting cells, they function like antigen-presenting cells, do they interact with these best 4+?
The best 4 cells all sit on the villi, and the M cells sit on the Paris patches. The best 4 cells, I don't show this here, I said they always sit next to a goblet cell. When we kill the best 4 cells, the goblet cells no longer can secrete their mucus. Right. Basically gets stuck and it goes to the wrong side, so they seem to be also important to control the right liquid situation for mucus to basically spread.
So basically
sort of in the ground state. Yeah.
Physiology of the gut, yeah. Very good. Yes.
Question. So really wonderful talk, Hans. You mentioned CFTR being expressed in best 4 cells. So when this channel is dysfunctional in the lung, the tissue becomes fibrotic and stiff. What happens with the gut? Is there any parallel in the effects between the 2 types of epithelium?
Yeah, so in Dutch actually the disease is called taai-slijmziekte, sticky mucus disease. And so when I did medical school, cystic fibrosis patients, which is some time ago, cystic fibrosis patients died of pancreas fibrosis. Chronic pancreas inflammation, because it essentially affects all of the tubes in our internal, so the gut, they have problems with passaging in the gut. The liver, bile gets stuck in the liver, but of course currently the biggest problem to get is in the lungs. But it's always the same thing, the mucus is too thick, and therefore the organ either gets clogged up, or in the lungs you get chronic inflammation, infection by Pseudomonas, for instance.
Go ahead.
So up here, it's a beautiful talk. I really enjoyed it. Actually, I wasn't familiar with the M cells until tonight. And you had mentioned that they differentiate due to signals from the underlying immune cells. So I'm wondering about your 3D models. It looked like the cells were being generated. And so do you have an immune cell co-culture there? Or how are the M cells being created at the baseline in your organoids without the Peyer's patch?
Yeah, so it was known, it was not the meiosis, it's an old observation that the rank ligand mutants don't have M cells, mice. And so, and we've published actually that mouse organoids have no M cells, but you give mouse organoids rank ligand, they make some M cells. There's a paper of a number of years ago. Same thing here, but humans often need more push than mouse organoids. So for the human organoids, we need rank ligand, which is produced by the Peyer's patches normally. We can just add it to the medium, you can buy it. And, but we also need this CSF2. And that actually cranks them up to 30-40% of the cells in the organoids are then M cells.
Thank you.
Hi. I had a question about the positional dependence of these cells. Sorry. I was interested in whether there are populations of M cells in certain locations more than others along the small intestine. And if there is, what could be the possible reason related to their functional importance?
Yeah. So they only sit on these Peyer's patches. And to be honest, I'm not really sure if the density of Peyer's patches is different along the axis of the small intestine. And they also sit in the colon. But so in mice, you're talking dozens of Peyer's patches. In humans, they tend to be small, and you have many more. But it's only the epithelium that grows from a crypt over a Peyer's patch that will have M cells. You never see them on villi or in crypts.
Thank you so much.
Normalizing screens and differentiations. I'm wondering, did you use other stem cells, like human pluripotent stem cells, iPSC, on your biogel or Invasin-coated plates?
No, so my lab never works with pluripotent stem cells, but we have done lung, pancreas, breast, human, mouse gut, snake venom gland actually also grows on the human growth factors and makes venom actually. So we think it's probably going to be work on, and as far as we can, and skin, and cornea and conjunctiva, so ectodermal, mesodermal, endodermal. So I think it's because it sees 4 or 5 different integrin complexes. It'll probably stimulate all epithelial cells. Now iPS cells I wouldn't know because they're not epithelial, but maybe if you grow them towards kidney and make a real epithelium or gut, you'll see the dependence.
I would like to finish with a question on your drug, cancer drug.
Oh.
Yes, so which is quite impressive. And this personalized medicine and personalized treatment of cancer is something that people are striving for. So in your studies, and particularly you said that this is undergoing a phase 3, so how do you select patients, and do you really stratify colon cancer in any way so that the compound actually works better than in some other types of cancer?
Yeah, so what you cannot do in a trial is stratify your patients on organoids and then use those patients in the trial. That is not, FDA would never accept that. So basically, but we know that it sees the EGFR and LGR5. So I'm not involved in these trials, so Meris runs these trials, but I know they at least record EGFR expression, but they don't select. So I think this is a so-called all-comers trial. It's actually head and neck, the one that they now have in phase 3. And the phase 2 results were spectacular. And they also think it's mostly because they can dose very high because it's so safe. And I would argue that the safety is because we actually screen for safety by showing no effect on normal organoids. And so we actually got like 50, 60 cancer drugs that are routinely used. And we're now testing them in the same setting, healthy versus for different, like 5 colon cancers, 5 breast cancers, 5 lung cancers. And what we see is essentially that almost all of these drugs kill the normal cells as well as they kill the cancer cells.
like chemotherapy, right? Thank you very much.